Today I will discuss about the complement system. The complement system is a part of the innate immunity. The complement system mainly synthesized by the liver and widely distribute the body tissue and body fluid. It is come into effect only when it is activated. There are 11 proteins are mostly involved, such as B, C1 to C9 and D. It is an enzyme precursor catalyzed series of the enzymatic reaction to product complement system. How it is activated? There are activated in three ways. Firstly, antigen antibody reaction, also known as classical pathway, and second is bacterial endotoxin, known as alternative pathway, and third one is lectin pathway, known as mannose lectin. The classical pathway is triggered by the antigen antibody reaction. Firstly, the C1 component of the complement system is activated and is converted into C4, and C4 component is converted into C4A and C4B, biggest molecule to form C14B, and C1 and C4B combine with each other and to form C2. Further it is converted C2A and C2B, and C14B and C2A is combined and form C14B2A. C3 converts C3A, it is convert C3A and C3B and to form C1462A. It is convert C5 and C5A and B. It's combined with 6, 7, 8, 9 and form membrane attack complex. And the C5A and C5A and C3B do not use in the pathway. It is used only other pathway. And next is alternative pathway. It is triggered by bacterial endotoxin. It is composed by lipopolysaccharide found in the outer membrane of the gram-negative bacteria, and second is cell wall and cobra venom factor. The first step of the spontaneous hydrolysis of the factor 3, which is present in a blood plasma. Factor 3 converts into factor 3C3B with the presence of H2O, and it combined with factor B. The factor B is converted factor BA and B presence of factor D, and it combined with factor C3B to form C3 convertase. Similarly, another pathway, C3B converted into C3B and C3A. It is convert C5 convertase membrane attack complex to form C5 B6, 7, 8, 9. The last one is lectin-mannose pathway. The lectin-mannose pathway binding lectin. It is present in Neisseria candidae and Salmonella. Whenever the organisms enter in the human body, the protein mannose binding recognize lectin these and activate the MASP mannose binding lectin associated serine proteins, which is bind MBL mannose binding lectin. It is triggered by the C4, C2, and their subtype is C4A and C4B. Similarly, C2A converts C2A and C2B. C4b and C2a combine and to form C4b C2a to form C3 convertase. And discuss about the immune effects. First is opsonization. The opsonization, when the C3b contact with microorganisms, they strongly activate neutrophils for the phagocytosis process of killing microorganisms. Next is cell lysis. It is a membrane attack complex C5b 6, 7, 8, 9. It ruptures bacterial cell wall. Next is chemotoxis. It is done by C5a attract neutrophil and macrophage for killing microorganisms. Next is activation of mast cell. It is done by C3a, C4a and C5a. It is activated mast cell and basophil, which is present their cause of large amount of inflammation, serotonin and histamine, which potential the immune system.